So we're going to finish up the course by talking about the regulation of gene expression. And we'll start off talking about how prokaryotes regulate the expression of their genes. And then we'll talk about a few things regarding eukaryotes. But eukaryotic gene regulation is much more complex than prokaryotic gene regulation is. So all living organisms, both prokaryotes and eukaryotes, will alter their gene expression in response to their changing environment. With prokaryotes, it's especially important they be able to rapidly adapt and change their gene expression because they're single-celled microorganisms. So they're all by themselves. Some eukaryotes are also single-celled organisms, but most eukaryotes are multicellular. So they live together with a bunch of other cells. In multicellular eukaryotes, gene expression regulates development and is responsible for the differences in cell types. So keep in mind that for an organism like you, a human, remember all of your cells have all of the exact same chromosomes, the exact same genes, the exact same alleles. Yet, a brain cell is not a muscle cell, it is not a heart cell, it is not a bone cell, it is not a liver cell. Even though they're all genetically identical, there are different genes that are active in those different cell types. That's what makes them a different type of cell, is they have different genes turned on and other genes turned off, right? So your muscle cells don't make bone, your brain cells don't make hemoglobin. There are certain genes that are turned on, others turned off. Certain genes that are always required, like the genes for cell respiration, those genes are always turned on. They're always being transcribed and translated. But all the other genes are only turned on and off as necessary. So all living cells have to regulate their gene expression. In prokaryotes, it's much simpler. Eukaryotes, it's much more complicated. As we're going to see, RNA molecules in eukaryotes play a huge role in regulating gene expression. So natural selection has favored bacteria that produce only the products needed by that cell. So remember, bacteria must be efficient. They cannot afford to be wasteful, right? Because they cannot rely on other cells, in a tissue or an organ to help them out. They're all by themselves. So have to be very careful in terms of what they make, when they make it, and how much of it they make. We've already learned about feedback inhibition, where the end product can feed back to an enzyme, an allosteric enzyme, early in the metabolic pathway, bind to the allosteric site, change the shape of the enzyme, which changes the shape of the active site, and turns that enzyme off, because now the substrate cannot bind. And because no product is made, there's no substrate for the next enzyme, and so on and so forth. So that's one way that the cell could regulate the formation of product, is by feedback inhibition. And we said the reason why cells did that was to save energy. But cells, and here we are going to focus primarily on prokaryotes, they can actually turn the gene off. So the enzymes do not get made in the first place. And remember, when we learned about translation and protein synthesis, a lot of ATP and GTP are used to synthesize a polypeptide. So protein synthesis is a very energy-intensive cellular process. By turning off the gene, those enzymes don't get made. That saves a lot more energy than feedback inhibition saves. So we learned about feedback inhibition. In this case here, tryptophan, an amino acid, is being synthesized by the bacteria. If they make too much, that tryptophan can actually feed back to an enzyme early in the pathway, bind to the allosteric site, turn the enzyme off, And so the cell temporarily stops making tryptophan. Remember, the binding of the tryptophan to the allosteric site is reversible and concentration dependent. So as the cell uses up the tryptophan, it's no longer available to bind to the allosteric site, so the enzyme goes back to its normal shape and begins to function, so that metabolic pathway gets turned back on, so the cell makes more tryptophan. We're going to learn that in prokaryotes, the end product, like tryptophan, can also feed back to the gene and turn the gene off so it's not transcribed, right? So messenger RNA is not made, and since there's no messenger RNA, right, there's nothing to translate to make protein. That saves way more energy than feedback inhibition does. And again, it's all reversible and concentration dependent. Now, prokaryotes, almost exclusively, they used to believe that using operons was exclusively prokaryotic. But as it turns out, there are some eukaryotes that do have some operons. But an operon is the functional unit to regulate gene expression in bacteria and other prokaryotes, like the archaea, that other major group of prokaryotes. And so what they do is they cluster all the genes needed for a metabolic pathway all together. They cluster them all together so they're all under the same control. So they're all turned on simultaneously. All the genes are turned off simultaneously. And that's very efficient. So instead of having one promoter for one gene, which is what happens most of the time in eukaryotes, in prokaryotes, you have one promoter for several genes. They're all lined up in a row, so that when the promoter starts transcribing, it transcribes all the genes needed to make all the enzymes for that particular metabolic pathway. So that's very efficient. You can turn on all those genes simultaneously, and you can turn them all off simultaneously. Most eukaryotic genes don't work that way. So there is a regulatory switch, a piece of DNA called the operator sequence, which is in between the promoter and what's called the structural genes. So the structural genes are the genes that actually code for the enzymes. So in between the promoter and the structural genes is the operator sequence. A repressor protein binds to the operator sequence, and it acts as a physical barrier to block the RNA polymerase. And the RNA polymerase simply cannot get past that repressor protein. So those genes will be turned off. So for prokaryotes, you have a promoter sequence, which is where the RNA polymerase binds. You have that operator sequence, and then we have all the structural genes. So all the genes that code for the different enzymes needed for that particular metabolic pathway. So a repressor protein can actually physically bind to the operator sequence and block it. So if RNA polymerase hops on and is trying to transcribe, it can't. It's physically blocked, right, by a repressor protein. It's a physical barrier. And so those genes will not be transcribed. Now, the regulatory gene is the gene that codes for the repressor protein. That's typically not located with the operon. It's usually some distance away on the chromosome, away from the operon itself. And the regulatory gene is under its own control mechanism. So a different control mechanism for the regulatory protein, for that repressor protein. So the operon can be switched off by a repressor protein. And that repressor blocks gene transcription because RNA polymerase cannot get past it. It prevents the RNA polymerase from being able to hop on and start moving, right, to transcribe. And the repressor protein is a product of a separate regulatory gene that we saw, usually located some distance away from the operon itself under its own separate control. But a repressor protein is there to help block the RNA polymerase. Now, repressor proteins can be in an active form or an inactive form. The active form is the form that's able to bind to the operator sequence. The inactive form does not bind, right, to that operator sequence. So in order to make the repressor protein active, there is often a co-repressor, a molecule that binds to the repressor protein to change its shape. So now the repressor protein can bind to the operator and turn off that operon. An example is the tryptophan operon in E. coli. And E. coli is a very common bacterium. It's in the guts of almost all mammals, including us. You're full of E. coli. And it's very easy to grow. As we talked about in lab last time, it's easy to grow, it's easy to manipulate. So E. coli has been extensively studied. So by default, the TRP operon is turned on, and the genes for tryptophan synthesis are transcribed. So there are certain operons that are in the on position and need to get turned off. So we call those repressible operons. So a repressible operon is one that's normally turned on. So it can be turned off, but it's normally turned on. So that's the default position, is being switched on. Because tryptophan is an important amino acid. It's needed for many proteins. And so the bacteria are normally synthesizing tryptophan because they need it almost all the time. But sometimes they either don't need it or they make too much. So too much tryptophan could turn off the metabolic pathway by feedback inhibition, by binding to the allosteric enzyme, or the tryptophan can act as a co-repressor, bind to the repressor protein, and together they bind to the tryptophan operon's operator sequence and turn off that operon. That way the enzymes for tryptophan synthesis stop being made. As the cell uses up the excess tryptophan, it no longer will bind to the repressor protein, which can no longer bind to the operator sequence, and then the operon gets turned back on. And we're going to see in more detail how that works. But I do want to mention an important difference between eukaryotes and prokaryotes. And this has to do with the messenger RNA synthesis. And we've already learned some important differences, right? Like eukaryotes have introns to be spliced out. Prokaryotes do not. Eukaryotes modify their messenger RNA with a 5' cap and a poly A tail. Prokaryotes do not do that. We mentioned how that it's slower for a eukaryote to synthesize a protein because transcription occurs in the nucleus and translation in the cytoplasm. And so the messenger RNA has to be transported from the nucleus to the cytoplasm. But in prokaryotes, there's no nucleus. So transcription and translation can occur simultaneously in a prokaryote. As that messenger RNA is being transcribed, a ribosome can hop on and start translating that messenger RNA even before it's finished being transcribed, right? Even before the mRNA is finished being completely made, it can start being translated by the bacteria. So bacteria can synthesize proteins faster. So here's another important difference: polycistronic versus monocistronic. So this refers to the type of messenger RNA. In prokaryotes, because all the genes needed for a particular metabolic pathway are all clustered together, one after the other, the RNA polymerase transcribes all those genes one after the other to create one large polycistronic messenger RNA. So what we see here is an mRNA which codes for those 3 genes. So the mRNA, it comes off as one big piece, one large messenger RNA, and then it will be translated as each enzyme, as each protein is translated from that messenger RNA, it will separate. And then as the ribosome continues to translate the next protein, and then it will separate. And then finally, that last protein, as it's translated, will be there. So one large messenger RNA transcript is translated as each protein or polypeptide is synthesized by the ribosome. It breaks off to be its own little separate, protein or polypeptide. Eukaryotes, with a few exceptions, do not do that. Eukaryotes have one messenger RNA for one gene. So that's monocistronic, one messenger RNA for one gene. But prokaryotes have a polycistronic messenger RNA. So one RNA with multiple genes. So that's just an important difference between eukaryotes and prokaryotes. Another important difference. And so once again, in eukaryotes, one messenger RNA for one protein. But in prokaryotes, one messenger RNA, which actually has more than one protein encoded on it. And as the ribosome translates it, all the individual proteins will separate. So that's just an important difference that I want you to know. Any questions on polycistronic versus monocistronic RNAs? So here we see the tryptophan operon. So we have several genes needed to code for the enzymes in the metabolic pathway that produces tryptophan. So this is a biosynthetic pathway, right, to make tryptophan. All those enzymes are clustered together under the control of a common promoter. So one common promoter. So when the tryptophan operon is on, all of these genes are transcribed simultaneously, one after the other. Then they'll be translated, right, from that long polycistronic messenger RNA. Each enzyme is going to come off and separate as it's being synthesized. There is an operator sequence. This is where the repressor protein binds. When the repressor is bound to the operator, it physically blocks the RNA polymerase and prevents it from being able to attach to the promoter and move past, right, to start transcribing. So right now the tryptophan operon is in the on position because that's the default position. It's a repressible operon, which is normally turned on, and then gets turned off when needed. So here we have our repressor protein. It is inactive, which means it cannot bind to the operator site, which is fine because most of the time the bacterial cell wants the tryptophan operon turned on, right, to make those enzymes to synthesize tryptophan, right? The cell wants that. But sometimes if it makes too much tryptophan or just doesn't need that much, it can turn the tryptophan operon off. Tryptophan acts as a co-repressor. It binds to the repressor protein to change the shape. Now the repressor protein can bind to the operator sequence and block that RNA polymerase. So then the operon is temporarily turned off, right? Tryptophan is important, and the cell's going to use it up. And once the cell needs to make more, then the tryptophan comes off of the repressor protein. We now have the inactive form of the repressor, which can no longer bind to the operator. RNA polymerase can hop on and start transcribing. So the operon has been turned back on. So a repressible operon is normally on, needs to get turned off sometimes, and then goes back to being on. But the end product, tryptophan, acts as a co-repressor to bind to the repressor protein, change its shape, so now the repressor can bind to the operator and turn off the operon. And so the cell temporarily stops making the enzymes needed to synthesize tryptophan. And so the cell is currently saving a lot of energy, right? Because translation, well, also transcription, but translation requires a lot of ATP, a lot of energy. And so the cell has temporarily stopped making those enzymes. Enzymes don't last forever, right? Proteins inside the cell don't last forever. The cell does not want them to last forever. That's one of the mechanisms of the regulation of gene expression, is that proteins and enzymes that are synthesized don't last forever because the cell may not need them, right, all the time. So here we see our active repressor protein now bound to the operator. RNA polymerase is stuck. It can't hop on the promoter. It can't get past the operator. So the operon is turned off, only temporarily. Remember, the binding of the tryptophan to the repressor protein is reversible and concentration dependent. So as the cell consumes the tryptophan, as it uses it up, there's less tryptophan to bind to the repressor protein. So eventually it comes off the repressor, and the repressor comes off the operator, and the operon is turned back on. So questions on the tryptophan operon? Normally turned on, needs to get turned off sometimes. So as I said, the TRP operon is a repressible operon. So this is an example of negative gene regulation because it's normally on and then it gets turned off. So that's a negative thing, right, to turn it off. An inducible operon is the opposite. An inducible operon is normally turned off, only gets turned on when needed. So repressible operons are normally for biosynthetic pathways. Inducible operons are for catabolic pathways. What's a catabolic pathway? What's catabolism? What's catabolism in catabolic pathways? All right, breaking something down to produce what? Energy, right? So an inducible operon usually codes for a catabolic pathway because the cell needs to find an alternate energy source, an alternate food source to make that energy. And so we have anabolism, right, for our repressible operons, anabolic pathways, biosynthetic pathways, and inducible operons are for catabolism or catabolic pathways. So as long as the cell has plenty of a particular food source, it's just going to use that particular food source. It's not going to turn on any other operons to use any other food sources unless it becomes necessary. And the reason why is turning on more operons, transcribing and translating more enzymes takes a lot of energy. If the cell already has an adequate food source, it doesn't need to turn on any other operons to use any other food source because it's got a food source, right? So turning on extra operons to eat some other food source when it's not necessary wastes energy. And prokaryotes are very efficient, right? They don't waste energy. They save energy. So the lactose operon, abbreviated lac operon for short, is an inducible operon. So it's normally turned off. And the reason why is because lactose, what is lactose again? You know a lot about lactose. Tell me about lactose. Where do you find it? What kind of sugar is it? What's it made out of? What kind of sugar is lactose? It's a disaccharide found in milk, right? So what are the two component monosaccharides, right, that make up that disaccharide? Glucose and galactose, right? So it's found in milk. It's found in dairy, right? So how common is lactose in the environment? I mean, look around the room where you are. Do you see lactose anywhere? So lactose is not very common. So the lactose operon is going to be turned off. It will only be turned on when absolutely necessary. So when is it absolutely necessary? Well, it's necessary when there's no more glucose available, right? Because glucose is the most common sugar, right? Glycolysis is a universal pathway. We know that prokaryotes, like bacteria, they can perform aerobic respiration or different types of anaerobic respiration like fermentation. But fermentation still uses glucose. So as long as there's plenty of glucose available, the lactose operon is going to stay off. It's only going to be turned on under certain conditions. So one of those conditions is the absence of glucose. All the glucose is gone. And the cell has to find an alternate food source. And it looks around, it's like, oh, there's lactose here. And then the cell will induce or turn on the lactose operon and start synthesizing the enzymes it needs to use lactose as an energy source. Now, inducible operons, there are molecules called inducers. An inducer molecule is what turns on the operon. So in this case, as we'll see, there is a repressor protein normally bound to the operator sequence. So that keeps the lac operon turned off. The inducer molecule is actually going to bind to the repressor protein and change its shape so it can no longer bind to the operator. So it's kind of the opposite of what we just saw with the trip operon, how it's normally turned off and a co-repressor, right, the end product will bind to the repressor protein and change the shape. So now the repressor can bind to the operator and turn off the operon. Here, the inducer is going to bind to the repressor protein and prevent the repressor from binding to the operator. In that way, the RNA polymerase is now able to get past and transcribe the genes needed for the cell to use lactose. So here we see the lactose operon. Again, there's a gene that codes for that repressor protein. It's normally located some distance away from the operon itself. We have our promoter sequence, our operator sequence, and then we have three genes which are encoded by the lac operon. Normally, the repressor protein is bound to the operator. Right? That's the normal condition, right? Because an inducible operon is normally turned off. It's only going to get turned on when absolutely necessary. So how does it get turned on? Well, allolactose, which for our purposes, is just lactose. It's slightly modified, but it's just lactose. So if the cell runs out of glucose, an alarm signal goes off inside the cell. And that alarm signal is cyclic AMP. So cyclic AMP is an alarm which also helps to stimulate the lactose operon to turn it on. But the idea, once all of the glucose is gone, an alarm goes off. It says, I'm out of glucose. You got to find something else to eat. And so the cell looks around, figuratively speaking. It's actually chemically sensing the environment to see what kind of molecules are out here that I might be able to eat. And if lactose is present, that will help to turn on the lactose operon, because lactose can bind to the repressor protein and turn the repressor protein off so it no longer can bind to the operator sequence. So as long as lactose is present and needed, it can bind to the repressor protein and inactivate that repressor. So now the operon is on. RNA polymerase can hop on and start transcribing these 3 genes. Now the 3 genes are beta-galactosidase, permease, and transacetylase. And the conversion messed up how this looks, but beta-galactosidase is the enzyme that splits lactose into glucose and galactose. So now the cell can use the glucose and the galactose, for energy. Permease is a transport protein which actively transports lactose into the cell. So permease upregulates the transport of lactose into the cell because the cell needs to use it now. And then transacetylase is not needed at all for cells to use lactose. And for many decades, scientists wondered why it was there, right? Why have this gene there if it's not needed for a cell to use lactose? Because you can knock out the gene for transacetylase, and bacteria like E. coli, they grow just fine on lactose. It's not needed. What they think the transacetylase does is that sometimes the lactose, remember if you think back to the labs with the Benedicts and the Barfage reagent, and we looked at the ring structures of the different sugars, well, there are different conformations of those ring structures. So they don't always fold in exactly the same way. And so there's basically a good ring structure and a bad ring structure. The bad ring structure makes that sugar toxic to the cell. And so they believe that transacetylase helps to detoxify the toxic forms of these carbohydrates if they occur, which is not very often because, like I said, the cells, they can use lactose just fine. They grow like crazy without the transacetylase gene. But there's some toxic conformations of carbohydrates, and the transacetylase is believed to help neutralize those toxic conformations. So that's why it's there. It's not really necessary, but it's a benefit, right? It does help to protect those cells. We will look at the animation shortly. So, as I said, the inducible enzymes or inducible genes, the inducible operons, they are catabolic operons, so they code for enzymes for catabolic pathways to break down a food source, use it to make energy. Repressible operons code for repressible enzymes, which function in anabolic pathways, which are biosynthetic. Normally, inducible operons are off. Repressible operons are on. And then an inducible operon gets turned on when needed, and then goes back to its off position when it's not needed. A repressible operon, it gets turned off when it's no longer needed, and then goes back to being on when it is needed. And so the regulation of the tryptophan and lactose operons involves both negative control and positive controls. It's actually a little more complicated than what we're going to go into. And these are just 2 examples of operons. Not all operons work this way, right? Most operons do have positive control mechanisms and negative control mechanisms. Some operons, like the arabinose operon, are really complicated, right? Because arabinose is not a common sugar. But remember, we used arabinose in the transformation experiment, right, to turn on our PBAD promoter so that the green fluorescent protein gene can be transcribed. And that was our example of gene regulation. And so in that plasmid P glow, we actually had two pieces of the arabinose operon spliced into that plasmid. One piece was the PBAD promoter, and the other piece was the ARAC gene. So the ARA is the arabinose. So those two pieces came from the arabinose operon. And we use them as a way to turn on genes. But the arabinose operon itself, I'm just mentioning it because its regulation is extremely complex, not the same as either lactose or tryptophan, because arabinose is not common at all. And so cells are only going to turn on the arabinose operon when absolutely necessary, right? There's nothing else the cell can possibly eat, and there is some arabinose present, then it will turn on that operon. But normally, it's hard to turn on the arabinose operon. Very complicated control mechanisms. So an example of positive regulation of the operons is CAP. So which stands for catabolite activator protein. So the CAP protein is a stimulatory protein. It helps to stimulate transcription. I've already mentioned the cyclic AMP. And so CAP is activated by binding with cyclic AMP. So cyclic AMP is the alarm signal. It can bind to the CAP protein, and then CAP gets activated. It can bind to the lactose operon and help stimulate it to get turned on. All right, so CAP attaches to the promoter, which enhances RNA polymerase binding. So CAP helps to stimulate induction of the lactose operon. But cyclic AMP activates CAP. But this is a positive feedback, right? Because it's helping to stimulate and enhance the lactose operon. So if glucose returns, if glucose becomes available again, then CAP will detach from the lac operon. And then eventually the lac operon will get turned back off. And CAP helps to regulate other operons as well. So here's just showing you cyclic AMP activates the CAP protein. Now CAP can bind to the promoter, which stimulates, attracts the RNA polymerase, helps it to bind to the promoter so that it can transcribe the lactose operon. Remember that lactose has to be present in order to bind to the repressor protein to change the shape, so that repressor can no longer bind to the operator. But once the cell has used up all of the available lactose is no longer available to bind to the repressor protein. Now the repressor will go back to its normal shape, bind to the operator, and turn off the lac operon. So lactose, well, all the glucose has to be gone first. Lactose has to be present in order for the lactose operon to get turned on, to get induced. The lactose operon is only going to stay on as long as there is sufficient lactose present. Once all the lactose is gone, the lac operon gets turned back off. Now the cell has to find something else to eat and turn on another operon to synthesize enzymes to use whatever that other food source is. And so bacterial cells constantly do this. If they have sufficient glucose, then they just use the glucose. If all the glucose runs out, it's all gone, then they have to turn on an operon to use something else. And that something else has to be present, like lactose. And that will help to induce the lac operon. But once all the lactose is gone, the lac operon gets turned off. Now the cell has to find something else to eat and turn on that operon. Some bacteria have a lot of different operons, use a lot of different food sources. E. coli is a carboholic. It loves carbohydrates, has a lot of different operons to use a lot of different carbohydrates. So E. coli will probably always be able to find some kind of carbohydrate to eat. But other bacteria are more limited, right? They can't use very many different carbohydrates because they don't have those operons. So they have to find something to eat, which they can eat, right? They have the ability to eat. Otherwise, they starve to death and die. So I do want to mention those genes that are so essential. They're always turned on. They're always being transcribed and translated. So like the genes for aerobic cell respiration. Since the glucose is the most common sugar, and you get the most ATP from glucose if you have oxygen available, those genes for those enzymes needed for glucose metabolism are always turned on. They're always being transcribed and translated. And so we call those genes constitutive. Right, so constitutive. They're always on. Right, they're never turned off because they're so essential. So much of the time, they're always on. They're always being transcribed and translated to make those enzymes. And so those are called constitutive genes. So they're so essential, so important, they're always on. Everything else is either a repressible or an inducible operon. So they either need to get turned on or turned off as needed. But constitutive genes, always on, always being transcribed and translated, right? Because they're so important. So questions on operons? Now we're going to watch the animation for lac operon. But any questions in general about the two operons we talked about? Lactose and tryptophan? So I'm going to share my screen so we can watch this animation. It's a Java animation, so it still works. So the two things that are floating around are RNA polymerase. Now, we're not showing the entire lactose operon. We're not showing the permease gene or the transacetylase gene. We're just going to show the beta-galactosidase gene. So we need a promoter for the gene that codes for the repressor protein. So the RNA polymerase is going to bind to that promoter. It will transcribe the gene for the repressor protein, which is magically going to be translated to make that repressor protein. So we'll watch this as it happens. So there's the messenger RNA. And this can be magically translated to make our repressor protein. So there's our repressor protein. And the repressor binds to the operator sequence. So I'll put the promoter here and the first gene for the beta-galactosidase enzyme. And so we notice that when RNA polymerase tries to bind to the promoter, it's blocked. It cannot get physically past, right, that repressor protein. And so the lac operon is turned off. but the repressor protein is bound reversibly, right, to the operator sequence. So every now and then that repressor protein will come off, and it's possible, and I'm going to remove this manually, it's possible the RNA polymerase is going to transcribe the lac operon a little bit. So there is a little bit of transcription of these inducible operons. So even though they're turned off, every now and then that repressor protein comes off and RNA polymerase can sneak in and transcribe. So, but very little of that happens. A little bit does, but very little. Right? So normally the repressor is bound to the operator. Right? So the RNA polymerase is stuck. Now, these are the beta-galactosidase enzymes, right? Because I allowed RNA polymerase to transcribe this gene a little bit. But so let's say all the glucose is now gone, right? The cyclic AMP alarm signal goes off. It binds to CAP, which helps to bind to the promoter to stimulate RNA polymerase to hop on and transcribe. But the cell needs to have lactose present in order for that to happen, right? Because lactose is able to bind to the repressor protein and change the shape of the repressor so it can no longer bind to the operator. So we have a lactose gun here. I'm going to fire off some lactose. There's some lactose being fired off. And so the lactose will bind to the repressor proteins and change their shape. Now they can no longer bind to the operator. So the RNA polymerase is able to hop on and start to transcribe the lactose operon. And there it goes. So now it's transcribing. And remember, it will transcribe all three genes, the beta-galactosidase gene, the permease gene, and the transacetylase gene. But we're only showing the beta-galactosidase. So that enzyme, when it's translated, is going to be able to cleave, to split the lactose into glucose and galactose so the cell can use them for energy. And there's the beta-galactosidase enzyme. So now it's going to start to attack that lactose and split it. So remember, the binding of lactose to the repressor protein is reversible and concentration dependent. So lactose will come off of the repressor proteins, then the beta-galactosidase will split it. Once all the lactose is gone, notice that the repressor protein has gone back and bound to the operator sequence to turn off that operon again. Because if there's no more lactose, there's no reason to keep on transcribing and translating the lac operon to make those enzymes if there's no more lactose present. At this point, the cell has to look for something else to eat and turn on another operon to use that alternate food source. So what we're seeing again right now is the normal position of the lac operon. It's in the off position because it is an inducible operon. It needs to be induced. It needs to get turned on. The way it gets turned on, all the glucose has to disappear. No more glucose to eat. And lactose has to be present. If lactose is present, it will bind to the repressor proteins, change their shape. Now the repressors can no longer bind to the operator. RNA polymerase is free to hop on and start transcribing so the cell can eat that lactose. But once all the lactose is consumed, then the repressor protein goes back to its normal shape and is able to bind to the operator and turn it off. So we're transcribing our beta-galactosidase gene. It will be translated to make the beta-galactosidase enzyme, which will start cleaving or splitting all the lactose. And there, translation occurs. Now the cell can actually eat that lactose. As the lactose is consumed, it no longer binds to the repressor protein, and the repressor will go back to its normal shape, bind to the operator, and turn off the operon. The cell's got to eat some more lactose. So now the cell has run out of lactose. The operon gets turned off. Now the cell's got to find something else to eat. So any questions on the lactose operon or tryptophan operon? I gotta find where I was before. Now, very interestingly, when it comes to E. coli, if you grow E. coli on glucose, you have a nice steep growth curve. So a nice steep growth curve, exponential growth. If you grow E. coli on just lactose, you also get exponential growth, but it's slower. Why is it slower? If it has plenty of lactose to eat, why is the curve not as steep as the curve with glucose? It's both exponential growth, it's just slower exponential growth with lactose than with glucose. So why do you think that is? That's a good thought question. Why would the growth be slower with lactose than with glucose? Yeah, it takes more time and energy to break down lactose, as Chris said. It takes more energy, right? Because you have to transcribe the lactoperon, you have to translate those enzymes. We know that takes a lot of ATP. So the cell has to spend a lot more ATP in order to make ATP from lactose. So the net return of ATP is lower with lactose. And so the cells don't grow quite as well. They still grow just fine, but not quite as well with lactose because they're spending a lot of ATP in order to use that lactose to make more ATP. And so your net return is lower, right? Because you've had to invest a lot more ATP to make ATP from lactose. With glucose, those genes are always being transcribed and translated, and they're still being transcribed and translated even when the lactoperon is turned on and there's no more glucose, because the genes for glucose metabolism are constitutive. So most of the time there's plenty of glucose available, but if not, cells will have to use something else. But using another food source is not as efficient. Does that make sense? And by the way, E. coli loves lactose, absolutely loves it. So that causes some problems in some people, right? Have you heard of lactose intolerance? Nobody's heard of lactose intolerance? Is anybody lactose intolerant? So human beings, they do not produce beta-galactosidase. human beings produce lactase. So that's the enzyme that humans use, lactase. But lactase is an enzyme that for many humans is only produced when they are a baby, because that's when humans naturally consume milk, right? Mother's milk, which contains lactose. And so we produce lactase. Many humans stop producing lactase after they are weaned, from mom, after they stop nursing from mom. And so if they try to eat a lot of lactose, like drink a big glass of milk, eat a big bowl of ice cream, they get sick, right? They get nauseous, they get bloating, gas, diarrhea, because the E. coli is having a field day. It's having a ball eating all that lactose. And that's what causes all the negative symptoms of lactose intolerance, because E. coli is loving it. Right, so many of the African peoples, many Asian people, they have difficulty with lactose because milk was not a normal part of their diet. Europeans, they started making things like cheese like 10,000 years ago or something. And so dairy became a common part of European diets. And so people of European descent, they now produce lactase constitutively. So they constantly produce lactase all throughout their entire lives. And that's actually an example of rapid recent evolution, where many Europeans, they now produce lactase constitutively. Those genes have been turned on and they stay turned on. But for many other people on the earth who did not, continue to consume dairy products, their lactase is dramatically downregulated. They just don't produce much, if any, lactase. So they have to limit dairy products. They may be able to eat a little bit without getting sick, but many people just cannot eat, milk or ice cream or cheese because it makes them too sick because they can't break down the lactose. You can buy some products where they break down the lactose before you consume it, so they add enzymes to break it down. Although those products are way more expensive, right? Because all the lactose has to be processed and broken down first. So I don't know what I would do if I became lactose intolerant, because I drink 1 or 2 gallons of milk a week, eat lots of ice cream, cheese. I don't know if I'd want to live if I couldn't eat lactose. So, as we already mentioned, all organisms must regulate which genes are expressed at any given time. So not all genes in an organism are always turned on. Some are on, some are off. Some get turned on and then get turned back off. Some are off and need to get turned back on, right? So it varies. When it comes to multicellular organisms, as I mentioned, gene expression varies from cell type to cell type. In single-cell microorganisms, we talked about how they have to be efficient. They can't afford to be wasteful because they're just a single cell. They don't have a lot of extra unnecessary DNA, which is why they don't have introns. And they normally just have the genes which are beneficial to them. Anything else that's not beneficial ends up getting lost from that population of bacteria because it's not beneficial. It's a burden. It's unnecessary. So microorganisms are very, very efficient. Now, I added a slide here just to clarify some things that for many decades, operons were exclusively found in prokaryotes until they found some operons in eukaryotes. So there are some eukaryotes that do have operons, like certain nematode worms like C. elegans and fruit flies, which are called Drosophila, which we talked about some of those early genetic experiments with the colors of their fruit fly eyes, because fruit flies have been a favorite organism for geneticists, forever. Some bacteriophages, right? Those viruses that infect bacteria also have operons. And there are eukaryotes whose ribosomal RNA genes are often controlled by operons. So you do find some operons in eukaryotes. It's not necessarily that widespread or common, but we do find some operons in eukaryotes. And when it comes to that polycistronic messenger RNA, again, almost exclusively prokaryotic, but there are some eukaryotic organisms, like some protozoan parasites, which have polycistronic messenger RNA transcription. So, as I've said before, there are always exceptions. So when it comes to operons, there are some eukaryotic exceptions. When it comes to polycistronic messenger RNA transcription, there are some eukaryotic exceptions, but that's not the common way it's done. Eukaryotes are mostly monocistronic, one gene, one messenger RNA. Eukaryotes have a variety of complicated mechanisms to regulate gene expression, but there are a few eukaryotes that have some operons. But those are the exceptions, not the rule. So when it comes to eukaryotes, there are many different ways to regulate the expression of genes in eukaryotes. And we're going to mention some of them. But because there are so many possible mechanisms of regulating gene expression, there are many possible places for mistakes to happen. And sometimes those mistakes lead to the formation of cancer. So remember that in eukaryotes, we have transcription, translation, mRNA modification, transportation to the cytoplasms for translation to occur. And then the proteins have to be modified. They may be glycosylated, the sugar may be added, right? They might be phosphorylated. They might have a fat added to them. There might be a lipoprotein. Then they have to be delivered, right, with the ER and the Golgi to deliver those proteins to wherever they're needed inside the cell. So there are every possible place from the gene all the way to the actual protein. Mistakes can happen, but also regulation can happen. And we're not going to go into all these different things, just a few of them. And we're going to focus first on the actual DNA itself. So we know that the DNA, those long linear eukaryotic chromosomes, are organized by wrapping around the histone proteins, right? That's what helps for those chromosomes to condense during mitosis, as they wrap around those histone proteins. And so we call that heterochromatin, because it's protein and DNA together, right? The chromatin and the heterochromatin is the DNA and the protein together, right? The histone proteins. If the DNA is very tightly wound around those histone proteins, if it's very compact, then it's normally not transcribed because it's so tightly, packed together. It's just not possible for the RNA polymerase enzyme to get in there and find a promoter and start transcribing. So very densely packed heterochromatin is usually transcriptionally inactive or silent. Now, there are some chemical modifications to the histone proteins which regulate the expression of the DNA. Histone acetylation is where acetyl or acetyl groups are attached to the histone proteins, specifically to the lysines, but they are attached to the histone proteins. That affects how tightly wrapped the DNA is. It actually loosens up the chromatin structure, which opens it up and allows RNA polymerase to get in, hop onto a promoter, and start transcribing. So this is another example of an epigenetic modification. So the genes, you may have those genes, you may have those alleles, they may not be expressed because those alleles are very tightly packed around the histones, and so transcription cannot occur. Even though you have that allele, it's not going to be transcribed. Or if your histones are acetylated, then the DNA is loosened up. Now RNA polymerase can get in and start transcribing. So another example of how you may possess a certain gene, a certain allele, it's not expressed, right? Because it may be turned off because your histones are not acetylated and the DNA is very tightly packed. RNA polymerase cannot transcribe. But if your histones do get acetylated, it loosens up the DNA, so now it can be transcribed. Another epigenetic modification? Well, if it's genetic, it's the actual DNA, right? The actual codons, right? That codes for a genetic trait. Epigenetic is a way to affect gene expression without changing the gene, without changing any codons. So you still have that gene, you still have that allele, but it's been modified either to be turned on or turned off, right? To be expressed or not be expressed. And that's called epigenetic. So if you had a mutation in that allele, it's not going to be expressed or expressed correctly, right? So you have a mutation, a possible genetic disorder, right? Because you're actually missing a functional enzyme. But in the case of an epigenetic modification, you have that allele and the allele is perfect. It's just been turned off. It's not being transcribed. So that's epigenetic, right? You still have that gene, you still have that allele. All the codons, all the nucleotides are all good. It's just that it's not being transcribed because of something like your histones not being acetylated. So does that help, Chris? So methylation of DNA is another epigenetic modification. So methyl groups can be added to the DNA. And so that causes the chromatin to condense. So methylation can often turn off a gene, so it's not transcribed. Phosphorylation, can do the opposite. It can loosen up the DNA so that it will be transcribed. So there's a lot of epigenetic factors which can affect the regulation of gene expression. Your histones may not be acetylated, which prevents the DNA from being transcribed. If your histones are acetylated, then your DNA can be transcribed. If your DNA is methylated, that turns off a gene, so it's not transcribed. If it's phosphorylated, that turns on a gene to help it get transcribed. So I don't know when exactly they started discovering these epigenetic modifications, but I'm thinking it was sometime in the 1990s. And so that's not that long ago. I know it is for some of you, but it's really not that long ago, the 1990s. And so again, it just helps to reinforce the idea that just because you have a particular gene, a particular allele, it doesn't mean it's going to be expressed. It's way more complicated than Mendel, right, ever could have imagined. And then we talked about DNA methylation in bacteria, which is different. The bacterial chromosome is methylated to protect it from those restriction enzymes, right? The restriction enzymes which cut up DNA, and they're used against bacterial viruses, right, to cut up their DNA to prevent infection. Well, methylation protects the bacterial chromosome, but it's not turning off the genes. in bacteria. So it depends on the organism as to what type of epigenetic modification exists and how it functions. So gene regulation is complicated. So here, just showing the histone proteins. Remember, the DNA, those long linear chromosomes need to be organized. And so they wrap around the histone proteins. If they're very tightly wrapped, there's no way that RNA polymerase can get in and start transcribing, so those genes are turned off. But if you acetylate the histone proteins, it relaxes the DNA, it opens it up, so now the RNA polymerase can get in and start transcribing it. So that's an epigenetic modification. And this has led to the histone code hypothesis. This proposes that specific combinations and modifications help to determine chromatin configuration, either tight or loose, and influence transcription. So all these things are happening inside of your cells. And so you may have epigenetic modifications, you may not. And so there are just so many places where genes get regulated in so many ways to regulate their expression. It's amazing that anything at all works correctly because there's just so many things going on, so many mechanisms of a gene regulation in eukaryotes, and every possible mechanism will cause a problem if it doesn't function correctly. So DNA methylation, where you add the methyl groups, is associated with reduced transcription in some species. So it's not consistent for everything, right? So as I said, with the bacteria, it does not affect their gene expression. But DNA methylation can cause long-term inactivation of genes in cellular differentiation. And this is actually something that is important that needs to happen, during our development. So when it's all done correctly, everything works right. If it's done incorrectly, that leads to problems and genetic disorders. And we mentioned genomic imprinting before, where you have cells where certain genes have been methylated, and it stays that way. So during the formation of gametes, the DNA gets temporarily unmethylated so that everything can happen properly during mitosis, but after the gametes are formed, those genes that were originally methylated, they get methylated again. So it's consistent. And it can vary, right? Whether it's maternal methylation or paternal methylation, right, to regulate gene expression. So it's epigenetic because the DNA is not being altered. None of the nucleotides are being changed. None of the codons are being changed. Everything is perfect and wonderful with regard to the nucleotide sequence. But whether or not that gene is going to be transcribed is what is being modified epigenetically. And most epigenetic modifications are passed on from parent to offspring. So epigenetic inheritance is something that, confuses the issue. It complicates the issue. It complicates the regulation of gene expression because sometimes, it's possible that in most of the offspring, a gene has been turned off, but for some reason in one of the offspring, it got turned back on. So if you've ever heard the expression of, a genetic characteristic skipping a generation or something, it may have something to do with epigenetic inheritance and whether or not it actually gets passed on, consistently. So there are enzymes, there's always enzymes that make all these things happen. So there are enzymes involved in all these things that modify the chromatin, right? You need an enzyme to acetylate the histone proteins. You need a different enzyme to methylate DNA. So there are enzymes involved in these epigenetic modifications. And that adds yet another place where something can go wrong. So transcription alone does not account for gene expression. There are many regulatory mechanisms which can operate at different stages of transcription, before, during, and after transcription. And these mechanisms allow the cell to fine-tune gene expression because, again, cells may need to change rapidly, because they have a rapidly changing environment. And so there are ways to adjust the expression, maybe to enhance the expression a little bit because it's needed, or to down-regulate the expression, right? Because you don't need to make as many of those enzymes. And we mentioned alternative RNA splicing before, because eukaryotic genes have introns, and the introns can be spliced out differently to produce different messenger RNAs, which are translated to make different proteins. So just to review that, a gene in eukaryotes, even though it's only one gene, it can actually code for more than one protein product as a result of alternative mRNA splicing. RNA molecules don't last forever, again, and that's a way to regulate the expression of a gene. You don't want the messenger RNA to be there and last a long time, because what if you don't need to make those enzymes anymore and you've turned off the gene, so you're not transcribing it to make any more messenger RNAs, but the ones that were already made, if they stayed active for too long of a period of time, then the cell ends up wasting energy and wasting resources. So mRNAs don't last all that long. Eukaryotic messenger RNAs last a little bit longer than prokaryotic messenger RNAs do because of that 5' cap and poly A tail on the eukaryotic messenger RNA, because it's got to be transported from nucleus to cytoplasm. So it does survive a bit longer, but in general, mRNAs don't survive that long, right? Because that's a mechanism of the regulation of gene expression. Now, when it comes to your actual DNA, you do have DNA that codes for some proteins, and DNA that codes for ribosomal RNAs, right, to make ribosomes, and a DNA that codes for transfer RNAs, right, to help with protein synthesis. But there's a lot of your genome that doesn't code for a protein, a ribosomal RNA molecule, or a transfer RNA molecule. It's not junk. It functions in the regulation of gene expression. Many different RNAs are transcribed. They don't get translated to anything, but the RNA molecules themselves act as regulators of gene expression. So we call these non-coding RNAs because they're not going to be translated into anything. But the RNA molecules themselves are involved in regulating the expression of other genes. We have microRNAs, miRNAs, which are small single-stranded RNA molecules, which combine to other messenger RNAs, which can prevent them from being translated by a ribosome, and they can help them get degraded. So again, a cell may not want to have that messenger RNA still be there, right, to still be translated when it doesn't need it. And so a microRNA can bind to that messenger RNA, stop it from being translated, and help that messenger RNA to be degraded. So a microRNA is a regulatory RNA. We also have interference RNAs, RNAis, and these are small interfering RNA molecules. And so those are siRNAs, but they interfere with gene expression. And so we actually have thought about using RNAs to block certain diseases, because if we can block the DNA or the RNA of that pathogen, either the DNA is not going to be transcribed or the RNA is not going to be translated. And so you've stopped the replication of that pathogen, right, by using these interfering RNAs or these microRNAs. The dicer enzyme is an enzyme which helps to produce these little interfering RNAs and microRNAs. siRNAs, the small interfering RNAs, they can affect heterochromatin, right? So again, that DNA wrapped around the histone proteins, some of these small interfering RNAs, they can actually block large regions of a chromosome from being transcribed. And they may also be specific for certain genes to block certain genes. So you're producing all these little RNAs, which are somehow modifying the regulation of gene expression, either by interfering with transcription, interfering with translation. And so you can imagine that if you're producing too many of these little RNAs, that actually starts interfering with gene expression. If you don't produce enough, you may have too much gene expression. So both of those situations can cause genetic disorders and diseases, including cancer, to occur. And then CRISPR, I remember you guys are supposed to be doing a discussion post on CRISPR. And so CRISPR stands for Clustered Regularly Interspaced Short Palindromic Repeats. We know what palindromes are, right? A sequence that reads the same way forwards as backwards. So these are repeated. And this was discovered in E. coli, right? Because we study E. coli a lot. And this is actually a defense mechanism against viruses. So it's part of a prokaryote's immune system. And there are two main CRISPR proteins that are used, or CRISPR enzymes, CRISPR-Cas9 and CRISPR-Cas3. But humans have started to use these to edit genes. And so we can use these for genetic modification in a human being, right? Because these enzymes, they can be specifically made to target a particular sequence of a particular gene. So if it's a defective or dangerous gene, those genes can literally be cut out of a chromosome and removed. So we use these enzymes for genome or gene editing. And so there are many ethical concerns about this. You know, in the old days, we talked about test tube babies, right? Selective breeding. Now we literally have the ability to edit genes, to actually change gene sequences in a chromosome. So that's something for you guys to do a little research on and find out that it has been done on humans. So you'll need to do some research on that and put it in your discussion post. And that's it. So last lecture is finished. Any questions or comments? Nope. All good. Okay. All right, so let's take a break. Let's come back to lab at 3:45 because lab is short, and then we'll do some reviews. So we'll take a good long break and then come back to lab about 3:45.
Thank you, Professor.
And then Jennifer, I will grade your lab exams as fast as possible, but those take time, so they may not be finished until Friday morning. And then I'll post, and then I have to calculate and post your tentative final grade so you can see whether or not the optionals can help you. I don't remember, Crystal. I'll check and see if I graded it. And you sent it back? When did you send it back? Let me stop recording.